Excretion of nitrogenous wastes. Excretion is the separation and elimination of metabolic wastes and the excess of essential materials from the body to maintain homeostasis. The cellular oxidation of non-nitrogenous compounds like carbohydrates and lipids produce non-nitrogenous wastes such as CO2 and H2. CO2 is eliminated by respiration, H2 enters the body fluids. It is eliminated only when present in surplus quantities. Oxidation of nitrogenous compounds like proteins, purines, etc. produce toxic nitrogenous wastes such as ammonia, NH3, urea, CH4OH, ON2, then uric acid, creatinine, etc. The accumulation of these wastes to toxic levels may disturb homeostasis. In addition to them, hormones, vitamins, and some essential ions such as hydrogen, sodium sulfate, phosphate, and chloride ions also may accumulate in excess of body's needs. The excess of these essential materials and toxic wastes are eliminated by the process of excretion. The nitrogenous excretory products of animals. The nitrogenous metabolic wastes are highly varied. The commonest ones among them are ammonia, urea, and uric acid. In addition to them, several minor products are also formed. They include creatine, creatinine, allantoin, then allantoic acid, and then hippuric acid, guanine, trimethylamine oxide, etc. The organisms in which they are formed are the following: ammonia, protozoans and most aquatic invertebrates including sponges, coelenterate, flatworms, crustaceans, marine bivalve mollusks, earthworms, freshwater bony fishes, amphibian larvae, etc. Urea, freshwater lamellibranchs, some teleosts, cartilaginous fishes, adult amphibians, mammals. Uric acid, terrestrial insects, terrestrial gastropods, reptiles, birds, man, apes. And creatine and creatinine, mammals. Allantoin, mammals, except man, apes, etc. Gastropods, dipterans, then allantoic acid, some teleosts. Then hippuric acid, mammals. Ornithinic acid, birds. Then trimethylamine, marine, bony, fishes. Then purines, various groups, guanine, spiders. Creatine is a nitrogenous acid synthesized in liver from an amino acid arginine, glycine, and methionine. It will be taken up by muscles and stored as phosphagen, creatine phosphate. Creatinine is the anhydride of creatine. It is a slightly water-soluble metabolic waste product. Allantoin is the end product of purine metabolism. Allantoic acid is a degradation product of allantoin. Both of these are excreted through urine. Hippuric acid and ornithinic acid are the products from dietary benzoic acid. Hippuric acid is formed by the combination of benzoic acid with glycine. Ornithic acid is formed by the combination of benzoic acid with ornithine. And types of excretion based on the chemical nature of the excretory products. 4 major kinds of excretion can be recognized among animals. They are ammonotelism, then ureotelism, guanotelic. Ammoniotelic animals are entirely aquatic. Ureotelic and guanotelic animals are exclusively terrestrial, and ureotelic animals include aquatic as well as terrestrial forms. Then ammoniotelic excretion. Ammonotelism is the excretion of nitrogenous wastes as ammonia. Animals which excrete ammonia are called ammoniotelic animals. They include aquatic protozoans, sponges, cnidarians, flatworms, leeches, crustaceans, marine lamellibranchs, freshwater teleosts, larval amphibians, etc. Flatworms, lungfishes, and metamorphosing amphibians are ammoniotelic as well as ureotelic. Crocodilians are ammoniotelic as well as ureotelic. Ammonia is produced by the deamination of amino acids. Deamination is the removal of amino group from an amino acid, producing free ammonia and corresponding keto acid by dehydroxy acid. It is of 2 major types, namely oxidative deamination and hydrolytic deamination. Oxidative deamination is the splitting of amino acid into ammonia and alpha keto acid. It is catalyzed by amino acid oxidase, or more precisely amino acid dehydrogenase. It is completed in 2 stages, namely oxidation and hydrolysis. Enzymatic oxidation of amino acid produces amino acid, which undergoes spontaneous hydrolysis and yields keto acid and ammonia. Hydrolysis of non-oxidative deamination is brought about by the deaminating enzymes amino acid hydrase or amino acid desulfhydrase. The former deaminates hydroxy amino acid, serine, homoserine, threonine, etc. And the later deaminates sulfur containing amino acids, cysteine, homocysteine, etc. The action of Dehydrasis is completed in 2 stages, namely dehydration and hydrolytic deamination. Dehydration produces an amino acid which undergoes hydrolysis and yields a keto acid and NH3. The action of desulfhydration is also completed in 2 stages, namely desulfhydration and hydrolytic deamination. Desulfhydration produces an amino acid which undergoes spontaneous hydrolysis yielding a keto acid and NH3. Ammonia is highly toxic and water soluble, so it has to be immediately eliminated from the body or converted to less toxic products such as urea and uric acid. Ammonia dissolve in water and form ammonium hydroxide NH4OH, which readily diffuses to the surrounding water. Thus, excretion of NH3 involves heavy loss of water and is possibly only in the water medium. So, ammoniotelic animals are aquatic forms. Then in terrestrial animals, loss of water has to be minimized or avoided. So in them, NH3 is not excreted as such. It is first converted to less toxic urea or uric acid and then excreted. Then uricotelic excretion. Uricotelism is the excretion of nitrogenous wastes as uric acid. The animals which exhibit uricotelism is called uricotelic animals. Then uric acid is produced from ammonia in liver and also by the metabolism of nucleic acids and purines. Uric acid is almost non-toxic and water insoluble, so it can be retained in the body for some time. It easily precipitates in crystals from a supernatural colloidal solution. Next is about ureotelic excretion. Ureotelism is the excretion of nitrogenous wastes as urea. Animals exhibiting ureotelism are called ureotelic animals. Urea is a water-soluble and less toxic. It is excreted through urine. Ureotelism is an adaptation for a semi-terrestrial life in which the animal is not necessarily surrounded by water. It is more advantageous than ammonotelism in that urea is much less toxic than ammonia, and so it can be retained in the blood for a long time. The elimination of urea requires only a small quantity of water, and so ureotelism minimizes water loss in excretion. Ureotelic animals include earthworms, prawns, cartilaginous fishes. Ureotelic animals include earthworms, prawns, cartilaginous fishes, frogs and toads, crocodiles, turtles, mammals, etc. Cartilaginous fishes can retain large amounts of urea in their blood. This makes their blood almost iso-osmotic to the medium and prevents osmotic loss of water from the body. Ureotelic animals in general excrete hypotonic dilute urine when there is heavy intake of water. When there is only small water intake, they excrete hypertonic concentrated urine. Then tadpole larvae of frog is ammonotelic because it is fully aquatic. At the same time, adult frog is ureotelic since it is amphibious. Lungfish is typically ammonotelic, but in summer when the ponds dry out, they may become ureotelic. Crocodiles are ammonotelic when they remain in water and uricotelic while they are on land. Earthworms are usually ammonotelic in the rainy season when there is sufficient water, but during dry season when there is only such much scarce water, they become ureotelic. Next is about human kidney. A pair of mesodermal kidneys are the principal urinary organs in man. In addition to them, skin also serves as an excretory organ. Human skins are bean-shaped and dark red bodies lying one on each side of the vertebral column. The inner margin of each kidney is concave and the outer margin convex. On the concave side, there is a depression known as hilus. From there leads down a duct called ureter. Terminally, ureter is open to the urinary bladder. Leading out from the bladder is a tube called urethra, short in female and long in male. In male, urethra joins the sperm duct and forms a urinogenital duct and opens out at the tip of the penis. But in female, urethra opens out independently. The opening of the bladder to urethra is guarded by a pair of muscular rings called urethral sphincters. This regulate the passage of urine. Urinary bladder is lined by transitional epithelium, which has great powers for distension and recoil or elasticity. It protects the bladder from damage due to enlargement when the bladder gets filled with urine. So inferior vena cava, aorta, adrenal gland, renal cortex, renal medulla, ureter, renal veins, renal artery, urethra, then urinary bladder is also there. Then close section of structure of the kidney. Each kidney is enclosed within a 3-layered fibrous capsule. The 3 layers of fibrous capsule are the inner renal capsule, middle adipose capsule, and the outer renal fascia. Renal capsule is a smooth, transparent and fibrous membrane. Adipose capsule is a mass of fatty tissue. Renal fascia is a thin layer of fibrous connective tissue. So medulla, capsule, cortex, callus, then renal pyramid, ureter. These are the regions in kidney. So some other portions: microstructure, loop of Henle, cortex, medulla, PCT, DCT, Malpighian capsule, collecting tubule, pelvis and ureter. So gross structure of kidney. Each kidney is enclosed within a 3-layered fibrous capsule. The 3 layers of the fibrous capsule are the inner renal capsule, middle adipose capsule and the outer renal fascia. Renal capsule, adipose capsule and renal fascia. Renal capsule is a smooth, transparent and fibrous membrane. Adipose capsule is a mass of fatty tissue. Renal fascia is a thin layer of fibrous connective tissue. Structurally, kidney has 2 regions, outer cortex and inner medulla. Renal cortex in turn has 2 zones, namely outer cortical zone and inner juxtamedullary zone. Cortex has several projections into the medulla known as renal columns or columns of Bertini. So renal medulla also has several conical bundles known as medullary pyramids or renal pyramids. Their apices are called renal papillae. Then cortex of the renal pyramid together constitute the parenchyma of the kidney. In the substance of the medulla, the base of the ureter expands to a cavity called renal pelvis. Its extensions, the spaces in between the renal pyramids, are known as calyces, singular calyx. And microstructure of kidney, that means structure of nephron. Kidneys are compound tubular glands. Each kidney is formed of about 1 million coiled tubules known as nephrons, renal tubules, nephric tubules, uriniferous tubules. These are the structural and functional units of kidneys. A nephron has 2 portions, namely Malpighian capsule and urinary tubule. Malpighian capsule is a basal capsular portion, then urinary tubule is a terminal tubular portion. Then Malpighian capsule in detail: Malpighian capsule, Malpighian corpuscle or renal corpuscle is the initial part of the nephron located in the cortical region. It has 2 parts, namely glomerulus and Bowman's capsule or glomerular capsule. Glomerulus is a vascular network. Glomerular capsule is a double-walled epithelial cup that encloses the glomerulus. The outer wall of the capsule is called the parietal layer and the inner wall is called the visceral layer. The space in between them is the capsular space. The parietal layer is formed of squamous epithelium and has no role in filtration process. The visceral layer consists of specialized epithelial cells called podocytes. So podocytes invariably rest on a basement membrane. They are flat cells with several marginal processes called pedicels. The narrow gaps between the pedicels serve as filtration slits for the passage of substances from glomerular capillaries to Bowman's capsule. Glomerular capillaries consist of highly porous endothelium. The visceral layer of the Bowman's capsule and the endothelium of the glomerulus together form the endothelial capsular membrane or filtration membrane. This membrane has extremely minute pores called filtration slits or slit pores for the ultrafiltration of water and solutes from blood to Bowman's capsule. So glomerulus is a capillary network. It is the center of the filtration of water and solutes from blood to Bowman's capsule. Blood enters it through an afferent arteriole, which is a branch of the renal artery. This arteriole breaks up into capillary network. Terminally, this capillary join together and form an efficient arteriole which carries blood away from the glomerulus, away in the sense, moving away from the glomerulus. Then moving away. Then efferent arteriole breaks up into a capillary network around the urinary tubule. This is called peritubular capillary network. It also carries to long loops of thin-walled capillaries called vasa recta. The capillaries terminally join together and form small venules which in turn join the renal vein. Then urinary tubule. Urinary tubule is the long and coiled part of nephron. It has 2 portions, namely proximal secretory part and distal excretory part. Excretory in the sense collecting part. Secretory part in turn has 3 portions, namely proximal convoluted tubule, distal convoluted tubule and Henle's loop. Convoluted tubules are the highly convoluted portions, contorted portions lying in the cortex region. Henle's loop is the narrow loop-like middle portion lying in the medulla. It has 2 limbs, namely ascending limb and the descending limb. Terminally, the excretory portion of several tubules join to form a collecting tubule. Several collecting ducts join to form a discharging tubule, duct of Bellini in the medulla. All the discharging ducts open to the pelvis at the summits of the renal pyramids. From the pelvis heads out the urinary bladder or ureter. Some portions are also there, like when we try to learn about the structure of Malpighian capsule, we could see portions like afferent arteriole, filtration slits, filtration membrane, podocytes, parietal layer, capsular space, visceral layer, efferent arteriole, glomerulus, renal corpuscle, Bowman's capsule, podocyte, squamous epithelium, and brush bordered cells. Microvilli also there. Structure of nephron in the sense, the whole structure. Renal cortex and renal medulla portions, afferent arteriole blood, Bowman's capsule, proximal convoluted tubule, efferent arteriole blood, glomerulus, distal convoluted tubule, excretory part, that means collecting tubule, thick ascending limb, thick descending limb, thick ascending limb, thick descending limb, thin ascending limb, and thin descending limb. Henle's loop, collecting duct, etc. are very important. Then, each limb of Henle's loop has two regions, thick and thin. The thick ascending limb is short and wide and impermeable to ions, urea, and water. On the other hand, the thin descending limb is freely permeable to water. The thin ascending limb is permeable to NaCl and urea. NaCl diffuses out of the limb and the urea diffuses into the limb. The two walls of proximal and distal convoluted tubules are formed by brush-bordered cuboidal epithelium with microvilli. The wall of the descending limb of the Henle's loop is formed by squamous epithelium, and the thin ascending limb consists of columnar epithelial cells, and that of the thick ascending limb is composed of flat cuboidal cells with numerous mitochondria and rudimentary brush border. These cuboidal cells are the sites of active outward transport of sodium and chloride ions. There are two types of nephrons, namely cortical nephrons and extramedullary nephrons. In the former, the glomerulus lies in the cortical zone and the remaining part of the nephrons clearly penetrate the medulla. On the later, glomerulus lies at or close to the junction between cortex and medulla, and the remaining part penetrates deep into the medulla. Then juxtaglomerular apparatus. As the afferent arteriole approaches the Bowman's capsule, the smooth muscle cells of its wall become highly modified. This nuclei become rounded and the cytoplasm become highly granular. Thus modified cells are called juxtaglomerular cells. Similarly, cells of the distal convoluted tubule adjacent to the afferent arteriole becomes considerably narrower. They are called macula densa. In addition to this, there are specialized cells between the afferent and efferent arterioles. They are called granular cells. The juxtaglomerular cells serve as mechanoreceptors for perceiving the pressure of blood in the arteriole. They also secrete the enzyme renin. Macula densa serve as chemoreceptors and they perceive changes in the solute concentration on the filtrate. The juxtaglomerular cells and the macula densa and the granular cells together form the juxtaglomerular apparatus. It plays a significant role in the regulation of renal blood pressure, control of renal blood flow, and the regulation of the reabsorption of chloride ions. It releases the enzyme renin, which mediates the conversion of angiotensin II by a plasma enzyme. Angiotensin II has a dual role. It raises renal blood pressure by increasing peripheral resistance and also promotes the secretion of hormones, aldosterone by adrenal cortex. Then functions of kidneys, function of nephrons. Apart from serving as the principal excretory and osmoregulatory organs, kidneys play several other roles also. The major functions are the following: filtration of waste products, especially the nitrogenous and sulfur containing products of protein metabolism, urea, uric acid, and from blood, maintenance of normal pressure, composition and volume of blood, maintenance of the normal osmotic balance or water balance in the body fluids by preventing the loss of water and at the same time removing the surplus water, maintenance of proper osmotic potential and body and tissue, then maintenance of proper ionic balance by removing the excess salts, regulation of acid-base balance and maintenance of normal pH by selectively removing acidic substances, and production and release of renin, erythropoietin and prostaglandins. Renin is the renal enzyme which mediates the synthesis of angiotensin, which in turn controls renal blood pressure and also promotes the serotonin secretion of the hormone aldosterone by adrenal cortex. Erythropoietin is the renal hormone which promotes erythropoiesis or the production of erythrocytes. Then regulation of blood sugar level under special conditions, activation and metabolism of vitamin D, synthesis of ammonia, hippuric acid and inorganic phosphates. These are some major functions. Filtration of waste products, especially the nitrogenous and sulfur containing products, maintenance of normal blood pressure, maintenance of normal osmotic balance, then maintenance of the proper osmotic potential in blood and tissues, maintenance of proper ionic balance along with regulation of acid-base balance, then production and release of renin, erythropoietin and prostaglandins, regulation of blood sugar level under special conditions, then activation and metabolism of vitamin D, synthesis of ammonia, hippuric acid and inorganic phosphates. Next is about urine formation in kidneys. The functioning of kidneys involves the formation and elimination of urine. Urine is a concentrated acidic solution of waste materials in water. The most widely accepted theory of urine formation is the secretion absorption theory. It holds that nephrons are the ultimate biological units of urine formation, and they exercise selective discrimination in the elimination or retention of various substances of the body fluids. Urine formation involves three major processes, namely glomerular ultrafiltration, tubular reabsorption, and active tubular secretion or augmentation. First process is glomerular ultrafiltration. This is the non-selective ultrafiltration or pressure filtration of water and low molecular weight substances from the glomerular blood to capsular space. Blood in the glomerulus flows under high hydrostatic pressure. This very high glomerular hydrostatic pressure forces out small molecules from glomerular blood to Bowman's capsule through the split pores or filtration slits of the filtration membrane. This is known as ultrafiltration or pressure filtration. This fluid thus filtered out is known as glomerular ultrafiltrate or nephrite filtrate or capsular filtrate. It forms the primary urine. Then its composition is almost the same as that of blood, except the blood cells and some plasma proteins are absent in it. It contains water, amino acids, glucose, vitamins, urea, uric acid, creatinine, sodium, potassium, calcium, chlorides, bicarbonates and other salts. The presence of blood cells and big high molecular weight proteins in urine indicate kidney disorders and defective glomerular filtration. Some driving forces of glomerular filtration. Glomerular filtration is mainly due to four driving forces, namely glomerular hydrostatic pressure, blood colloidal osmotic pressure, capsular hydrostatic pressure, and net filtration pressure or effective filtration pressure. GHP is created when blood in the glomerulus flow under high tension from wide afferent arterioles to narrow afferent arterioles. BCOP is mainly due to the concentration of blood plasma proteins, particularly albumins. CHP is the pressure of the fluid in the Bowman's capsule. EFP, also called filtration pressure, is the difference between GHP and the sum of BCOP and CHP. So EFP is equal to GHP minus summation of BCOP plus CHP, which are the glomerular hydrostatic pressure GHP, blood colloidal osmotic pressure BCOP, capsular hydrostatic pressure CHP, and net filtration pressure or effective filtration pressure EFP. GHP tends to dry water and dissolved substances from blood to Bowman's capsule. On the other hand, BCOP and CHP have an opposite effect. They tend to draw fluid substances to blood. The glomerulus GHP is approximately 70 mmHg, BCOP is 30 mmHg, and CHP is 20 mmHg. So the net filtration pressure is approximately 20 mmHg. Then next step is tubular reabsorption. This is the selective reabsorption of useful substances from the glomerular filtrate back to blood. Tubular reabsorption occurs to the epithelial cells all along the renal tubule. Water, glucose, amino acids, vitamins and some ions such as Na+, K+, Ca2+, Cl-, HCO3-, HPO4-, etc. are reabsorbed by this process. This helps the body to retain most of its chemical constituents. About 99% of glomerular filtrate is reabsorbed. So only about 1 to 1.5 liters per day will be excreted as urine. Tubular reabsorption is a selective process because some substances are reabsorbed in large quantities, some in very small quantities, and still others are not at all reabsorbed. The selective reabsorption is based on body's needs. The maximum amount of substance that can be absorbed under any condition is called the tubular maximum of that substance. There is an optimum rate of renal reabsorption for every substance, which is called the renal threshold. It is different for different substances. Normally, glucose and amino acids are almost completely reabsorbed. Water, vitamin C, Na plus, and some salts are reabsorbed partly or completely depending on their concentration in blood. Urea, uric acid, etc. may not be reabsorbed or may be reabsorbed in negligible amounts. Creatinine, sulfates, uric acid, etc. are not at all reabsorbed. The substances that are reabsorbed in large amounts is called high threshold substances. Glucose, amino acids, vitamin C, water, Na plus, C plus, Ca2 plus, etc. Those which are reabsorbed in very small amounts are called low threshold substances, and substances which are not at all absorbed or reabsorbed are called non-threshold substances, sulfates, creatinine, hippuric acid. Tubular reabsorption involves both active and passive transport mechanisms. Water and some salts are reabsorbed by passive transport in a concentration gradient. On the other hand, glucose, amino acids, Na plus, K plus, Cl minus, Ca2 plus, etc. are reabsorbed by active transport against concentration gradient in energy with energy expenditure. Reabsorption of water and dissolved constituents occurs mostly in the proximal convoluted tubule. The fluid left out after this is known as the secondary urine. It slowly passes along the tubule. When it reaches the distal convoluted tubule, final reabsorption of water and sodium occurs. Then tubular secretion, augmentation. This is the active secretion of some substances from blood to urinary tubule. It occurs in the distal convoluted tubule. H plus, Na plus, K plus, ammonia, sulfur compounds, hippuric acid, creatinine, steroids, organic acid, and reactive drugs such as penicillin, etc. are secreted by this process. Tubular secretion is relatively less significant in glomerular kidneys but most significant in agglomeralar kidneys. Tubular secretion serves two purposes: remove some substances from the body, especially K plus and H plus, then conserves base composition and controls blood pH. After selective reabsorption and active secretion, a concentrated and acidic solution of water is left in the tubule. This is called final urine. The secretion of H plus and ammonium ions give the urine an acidic pH of 6. Then some aspects related to urine concentration. Mammals and birds are capable of producing highly concentrated urine which is hyperosmotic, hypertonic to blood. This ability depends upon the following factors, like presence of loop of Henle and its relation to collecting tubule and vasa recta, and close proximity of the ascending and descending limbs of Henle's loop and the difference in their permeabilities to water and solutes. Descending limb is permeable to water and impermeable to solutes, whereas the thin ascending limb is permeable to solutes. The active transport mechanism operating in the thick ascending limb. Operation of an endogenous mechanism called countercurrent mechanism in Henle's loop and vasa recta. High osmotic concentration in the peritubular interstitial fluid around the Henle's loop. An osmotic gradient between peritubular fluid and intratubular fluid. Loop of Henle is highly specialized for urine concentration. It is long up in juxtamedullary nephrons, especially in those mammals which must produce highly concentrated urine for water conservation. The larger the loop of Henle, the more concentrated would be the final urine. Also in such animals, there exists a strong osmotic gradient which causes the osmotic flow of water from the less concentrated hypotonic tubular fluid to the more concentrated hyperosmotic peritubular fluid to make the extending urine highly hypertonic. Next is about countercurrent mechanism or urine concentration. This is the physiological mechanism responsible for the high osmotic concentration of urine and peritubular fluid. In this mechanism, loop of Henle acts as a countercurrent multiplier system and vasa recta as a countercurrent exchange system. Henle's loop and vasa recta are in the form of shape of a hairpin loop with a descending limb and an ascending limb. So tubular fluid, glomerular filtrate in Henle's loop and blood in vasa recta flow in opposite directions in the two limbs, downward in the descending limb and upward in the ascending limb. This may be called a countercurrent flow. Since the two limbs are close to each other, exchange of materials is possible between them to regulate the osmolarity of their fluid contents. This is called countercurrent exchange. Countercurrent mechanism involve three factors at work. They are active transport of solutes by the thick ascending limb of Henle's loop, maintenance of differences between the ascending and descending limb with respect to their permeabilities to water and NaCl, and U-shaped configuration of the Henle's loop which facilitate a countercurrent flow of the tubular fluid. So countercurrent mechanism involves three factors at work. They are active transport of solute by the thick ascending limb of Hell-Needs loop, maintenance of the differences between the ascending and descending limbs with respect to their permeabilities to water and NaCl, and U-shaped configuration of the Henle's loop, which facilitates a countercurrent flow of the tubular fluid. The descending limb of Henle's loop is permeable to the osmotic outflow of water and the passive inward diffusion of NaCl, although it does not actively transport NaCl. So, in it, water flows outward from the less concentrated glomerular filtrate to the more concentrated peritubular fluid, and NaCl diffuses inward in the opposite direction. Consequently, the glomerular filtrate gets progressively more concentrated by the addition of more and more NaCl while it flows down along the descending limb. However, its concentration will not exceed that the peritubular fluid around Henle's loop. The ascending limb of Henle's loop is permeable to water, but it serves as a site for an active outward transport of NaCl from glomerular filtrate to peritubular fluid. This active transport results in an increase in solute concentration or osmolarity in the peritubular fluid, and thereby establishes a strong osmotic gradient between the peritubular and tubular fluids. The glomerular filtrate becomes progressively less concentrated or less osmolar by losing NaCl as it flows upward in the ascending limb. When the flow of fluid in the ascending and descending limbs of Henle's loop is in opposite direction is called countercurrent flow. Thus, the two limbs constitute a countercurrent flow system. The active and passive movements of NaCl in the system produce an osmotic gradient. At any one place in the Henle's loop, the concentration difference between the contents of the two limbs is only moderate, but along the length of the loop, this moderate difference is additive and it has a multiplication effect. Therefore, the entire countercurrent flow system is called a countercurrent multiplier system. The final fluid concentration in this current system is determined by the length of the loop, and longer the loop, the higher the urine concentration. Thus, there is a direct correlation between the differential relative length of the Henle's loop and the maximum concentration of urine. And the collecting tubule is a variably permeable to water. Its water permeability depends upon the relative concentration of the peritubular fluid. If the peritubular fluid is more concentrated, hyperosmotic, than the fluid in the collecting tubule becomes water permeable and water escapes from it by osmosis. This results in a highly concentrated urine. If the peritubular fluid is isosmotic or hypoosmotic to the fluid in the collecting tubule becomes impermeable to water and no water is lost from it or gained by it. This results in the production of dilute urine. Concentrated urine is produced when the fluid volumes in the body is low and water has to be conserved. Dilute urine is produced when the fluid volume in the blood is high, body is high, and the excess water has to be eliminated. Concentrated urine is produced in very small quantities, but dilute urine is produced in large quantities. The glomerular ultrafiltration that enters the proximal convoluted tubule during countercurrent flow is almost isosmotic to the peritubular fluid with an osmotic concentration of approximately 300 milliosmoles. By the time it reaches the bend of Henle's loop, it becomes increasingly more concentrated, hypertonic or hyperosmotic, with an osmotic concentration of about 1200 milliosmoles. As it moves up along the ascending limb, it becomes progressively less concentrated, and by the time it reaches the distal convoluted tubule, it becomes isoosmotic or hyperosmotic, with an osmotic concentration of nearly 100 mmosmoles. Again, in the collecting tubule, it becomes progressively more concentrated if water has to be conserved, or becomes hypoosmotic if water has to be eliminated. Then it becomes clear that two major events occur as glomerular filtrate passes along the renal tubule. They are osmotic outflow of water from the descending limb of Henle's loop and the collecting tubule, cyclic movement of sodium in which sodium enters the descending limb of passive diffusion and escapes from the ascending limb by active transport. These two processes operate simultaneously, one facilitating the other. As a result, urine becomes progressively more concentrated in the descending limb and collecting tubule and progressively less concentrated in the ascending limb. Normally, the excreting urine is concentrated. However, under abnormal conditions, the collecting tubule becomes impermeable to water, with the result that excreting urine is dilute. The outflow of sodium from the ascending limb of Henle's loop makes the tubular fluid hyperosmotic and the peritubular fluid hyperosmotic in relation to each other. This in turn increases the osmotic potential of the peritubular fluid and creates an osmotic gradient that causes the osmotic outflow of water from the descending limb and the collecting tubule. Water cannot flow out from the ascending limb since it is impermeable to water. The outflow of water makes the tubular fluid and is in the descending limb and collecting tubule hyperosmotic to the peritubular fluid. Consequently, the osmotic potential of the peritubular fluid falls low. This causes the outflow of sodium from the ascending limb and the cyclic repetition of the whole series of events. The functional state of the countercurrent multiplier mechanism is maintained by establishing an osmotic gradient between the peritubular and tubular fluids. This osmotic gradient determines the direction of the movement of water and solutes. It is established by the countercurrent exchange mechanism of vasa recta, which causes the movement of water and or solutes without allowing them to accumulate anywhere in the loop of Henle. So vasa recta extends deep into renal medulla parallel to the loop of Henle. The blood entering its descending limb is hypotonic to the interstitial fluid and its osmotic concentration is about 320 mMol. So as it flows down to the renal medulla, water passes out from it to the interstitial fluid by osmosis and NaCl gets into its interstitial fluid by diffusion through the capillary wall. As a result, the osmotic concentration of the blood in the ascending limb progressively increases. When this blood flows upward to the renal cortex along the ascending limb of vasa recta, NaCl escapes from it by diffusion and water enters it by osmosis. This vasa recta and countercurrent exchange aid in maintaining the osmotic concentration gradient of the interstitial fluid, essential for maintaining the functional state of the countercurrent multiplier mechanism of Henle's loop. Next is about chemical composition of the human urine. Normal human urine is formed of water, nearly 96%, organic waste 2.5%, and inorganic salts 1.5%, and trace amounts of other elements. The major organic constituents include urea, uric acid, hippuric acid, creatinine, ketone bodies, oxates, indican, etc. So inorganic constituents include NaCl and chloride, sulfates and phosphates of potassium, calcium, magnesium and ammonium, and sulfate and phosphate ions. Urea is the most abundant organic compound and NaCl is the most abundant inorganic salt. Then regulation of kidney functioning. Urine formation is regulated by the sympathetic nerve system. Moderate stimulation of the sympathetic nerve causes the constriction of the afferent glomerular arteriole and thereby lowers the glomerular filtration rate. The GFR is mainly influenced by the colloidal osmotic pressure of the plasma in the afferent glomerular capillaries. Most of this plasma get filtered in the glomerulus, raising the colloidal osmotic pressure in the glomerulus. This rise in colloidal osmotic pressure in all probability prevents or lowers glomerular ultrafiltration. In brief, sympathetic stimulation constricts afferent glomerular arterioles, raise colloidal osmotic pressure in glomerulus, reduces glomerular ultrafiltration and lowers GFR. Dilation of the afferent glomerular arteriole increase the GFR. A very strong sympathetic stimulation has a dual effect. On one hand, it causes the constriction of afferent glomerular arterioles and a rise in glomerular pressure. This may increase the GFR. On the other hand, it causes a decrease in the rate of blood flow through glomerulus and consequent rise in glomerular plasma colloidal osmotic pressure. This may decrease the GFR. Thus, a very strong sympathetic stimulation simultaneously lowers and raises the GFR. These two effects may cancel each other so that GFR may remain unchanged. Then hormonal regulation. The major hormones involved in the regulation of urine production include aldosterone or adrenal cortex, epinephrine, adrenaline of adrenal medulla, vasopressin, antidiuretic hormone ADH of hypothalamus, thyroxine of thyroid gland, parathyroid hormone, and the atrial natriuretic hormone produced by some cells of the atrial wall. Aldosterone stimulates the tubular reabsorption of sodium and the tubular secretion of potassium. Epinephrine inhibits glomerular filtration and thereby decrease urine production. Vasopressin reduces water loss through excreting urine. It promotes the tubular reabsorption of water and thereby makes the excreting urine highly concentrated and so decreases the quantity of final urine. Low water content in the body fluid promotes its production and high water content inhibits its production. So deficiency of ADH may cause diabetes insipidus. It is characterized by inadequate renal reabsorption of water and loss of large quantities of water through hypotonic urine. Thyroxine reduces tubular reabsorption of water and thereby increases diuresis, renal output of urine. Then parathyroid hormone is a calcium regulating hormone. It promotes the tubular reabsorption of calcium and magnesium and inhibits the tubular reabsorption of phosphates. An atrial natriuretic hormone, atriopectin, inhibits the tubular reabsorption of water and sodium and thereby increase their urinary excretion. It also lowers the secretion of aldosterone and this increase indirectly promotes the tubular reabsorption of sodium and potassium. Then enzymatic control, renin-angiotensin system. The enzymatic control of urine production is accomplished by renal enzyme renin. It is a proteolytic enzyme produced by the juxtaglomerular cells of the kidney. It mediates the conversion of liver protein angiotensinogen to angiotensin. Angiotensinogen, also called a renin substrate, is an inactive circulating globulin of more than 400 amino acid residues. It is produced in liver. Renin converts it into another inactive form called angiotensin 1, which is a decapeptide formed of 10 amino acid residues. Angiotensin 1 is soon converted to an active form called angiotensin 2, formerly called hypertensin or angiotensin, by the enzyme angiotensin converting enzyme. Angiotensin 2 is an octapeptide formed of 8 amino acid peptides residues. It is soon converted to angiotensin 3 by the enzyme aminopeptidase. Angiotensin 3 is a heptapeptide formed of 7 amino acid residues. And angiotensin 1 does not have any physiological function. It simply serves as a precursor of angiotensin 2. On the other hand, angiotensin 2 and 3 have definite physiological roles. They serve as vasoconstrictors and thereby elevate systolic and diastolic blood pressure. This in turn promotes glomerular ultrafiltration, especially the filtration of sodium. Also, they stimulate the production of aldosterone, which in turn stimulates the tubular reabsorption of Na and tubular secretion of K. Abnormal constituents of human urine. Sometimes traces of substances that are not usually present may appear in urine, or normal constituents may appear in abnormal amounts. For example, several unusual substances taken into blood or formed within the body are got rid of through urine. This is mainly due to effective glomerular ultrafiltration and tubular reabsorption. A clinically important abnormal constituents of urine include albumin, glucose, ketone bodies, blood, bile pigments, pus, and tube casts, etc. Albumin. Proteins in general are giant molecules, so they cannot normally pass through the split pores of glomerular filtration membrane during glomerular ultrafiltration. Still, under unusual conditions, they pass out and then make their appearance in urine. The presence of proteins in the final urine is known as proteinuria. Albumins, globulins, and fibrinogen are the blood plasma proteins sometimes found in urine. Normally, albumin is absent in urine since glomerular walls are impermeable to it, but in persons with kidney disorders, large quantities of it pass from glomerular blood to Bowman's capsule and then show up in urine. The presence of albumin in urine is known as albuminuria. So, prolonged albuminuria naturally leads to abnormal reduction in plasma proteins. So, albuminuria is clinically important in that it is often a symptom of serious heart disease or kidney disorder. Albuminuria mostly result from non-selective permeability of glomerular capillaries and bleeding in nephrons. The common cause of albuminuria are kidney disease such as nephrosis, then glomerular nephritis, acute and chronic infections of kidney, bladder, and urethritis or cystitis, then pyelitis, heart disease, prolonged hypertension, fever, anemia, etc. Some drug and toxic quinine, arsenic, salicylic acid, lead, mercury, gold, etc. may also cause albuminuria. So, transitory forms of albuminuria are occasionally found in healthy normal person also. For example, orthostatic or postural albuminuria is sometimes found in persons who spend long periods in a standing posture. So, athletic albuminuria is found among athletes during periods of rigorous practice and severe muscular exercise. So, adolescent albuminuria is found among growing boys. Apart from this, albuminuria sometimes become occur in some persons after cold bath after eating large quantities of protein-rich food or raw eggs. And glucose. Glucose is one of the major constituents of glomerular filtrate. In normal kidneys, it is almost completely reabsorbed in the renal tubule, so long as the level of blood glucose is not abnormally high. So glucose will not be present in the final urine, but in some instances, blood sugar level goes very high, hyperglycemia. Consequently, renal reabsorption of glucose decreases or stops. As a result, glucose gets heavily concentrated in urine. The presence of large amount of glucose in excreting urine is known as glycosuria. So prolonged and persistent glycosuria is associated with diabetes mellitus or sugar diabetes, in which cellular uptake and utilization of glucose is very low, mainly due to deficiency or absence of pancreatic hormone insulin. Then blood cells may be present, bile pigments, casts. Casts in the sense, casts are tiny masses of hardened materials shaped like lumen tubules. They are formed within cavities in the body and then flushed out. Casts are named after the materials which form them. Examples are leukocyte casts, erythrocyte casts, epithelial casts, granular cell casts formed by the degradation and decomposition of cells, fatty casts formed by the fatty cells, and the presence of tube casts. The casts of renal tubules in urine is a sure sign of glomerulonephritis. Then is about some major renal disorders: renal failure, hematuria, uremia, renal hypertension, nephritis, nephrosis, and renal calculi. Then edema and acidosis are the alkylosis. These are the major diseases. Alkylosis or alkalemia is the opposite condition of acidosis. It is characterized by low hydrogen ion concentration of arterial blood in it. Blood pH is above 7.45 due to accumulation of bases or loss of acids in the absence of compensatory changes. Acidosis or acidemia is the abnormal clinical condition in which the body pH is below 7.35, usually between 7.35 and 6.8, either due to accumulation of acids or due to the loss of bases in the absence of compensatory changes. In severe acidosis, total body sodium gets mostly depleted, plasma sodium levels falls low, level of total body potassium becomes low, and the level of plasma potassium remains almost normal. In acidosis, there is a production of two abnormal acids, namely acetoacetic acid and beta hydroxybutyric acid, and there is a fall in the alkali reserve of the body. The production of abnormal acids usually results from the faulty metabolism of fats. Acidosis occurs mostly in cases of untreated and partially treated diabetes mellitus, starvation, persistent vomiting, and detailed anesthetic vomiting. It may also occur in the terminal stages of glomerulonephritis. A mild form of it may occur in severe fevers, especially in children. The common symptoms of acidosis include general lassitude, severe vomiting, increased thirst, restlessness, and the presence of acetone in urine. There are two major types of acidosis, namely respiratory acidosis and metabolic acidosis. The former is characterized by abnormally high PCO2.